phenotype maps in the sense of a genome-wide association study. And I study the fly wing as a model system. And the basic conclusion of this talk is that it makes no sense to think about an association problem as a univariate, or a series of univariate properties. And this is brought home particularly strikingly for the case of morphometric variation, where you literally cannot change the location of a point on a morphological structure without changing other things about that morphological structure. It is impossible. So if you were picking a whole series of sort of unrelated traits that you could imagine were genetically independent of one another, it might make sense to think of them as, oh, I wonder if this one has, I wonder if this one has, I wonder if this one has. But really, most things on organisms, say the metabolome, you literally cannot change one element there without having impacts elsewhere. So again, the univariate approach just doesn't make sense. So just to give you a little bit of an example, I'm sorry, the color is terrible here. There's a green arrow. I'm going to say that's our trait, okay, a univariate trait. And we have a SNP that affects it and makes that a little bit shorter. And here's another SNP that has exactly the same effect on that one measurement. But it makes a huge difference whether what it's doing is this versus that, to have that same effect. So treating that as one effect on everything is, to me, the sensible way. So here's our data set. This is from the Drosophila Genome Reference Project. We measured 184 of those lines. They were independently measured in 2 different labs. The repeatability is excellent. And we chose to take the representation out to so 49 points here. But we actually fit these functions to the vein, so we could have gone to twice as many if we wanted. That makes things, computers have to work harder. And here's the aligned data once it's all put together. So let me just go back and talk about, well, the usual univariate approach. So let's say we measured 2 things, say the length and the width of the wing, and then we have 2 genotypes. And in this case, you can just look at it and say that, well, they differ in both of them. So the univariate analysis, you're essentially projecting on one axis. They're different. You project on another axis. They're different. That's great if you have separation on both of those axes. But here's another case where we have an effect that you can clearly see, but if you project on either of the width-length axes, you would not see a difference. And this is the essential advantage of a multivariate statistical approach to the problem. And geometrically, this is sort of what happens. You find the direction in which they differ the most by some criterion, and then you're projecting the data on essentially an axis that you define. And that direction is the trait that is affected by. So we're letting the SNPs, the effects of the SNPs, the inferred effect of the SNPs, define what a trait is. And every SNP, of course, could affect a different trait in that sense. So here's the mystery. It's like, okay, to me this is really obvious, and it's probably because I work on this sort of integrated structure where you can't change one thing without changing another. But if you look in the literature, there's almost nothing multivariate in the whole association field. And in some cases, this is due to the fact that, well, in human studies, they don't measure everything on everybody. And so there's other problems there. But we should really be doing this. So why don't we do it? Well, I get the sense that there's anxiety. Nobody's doing it. There must be a reason why nobody's doing it. So it's the curse of dimensionality. But if you ask people, what is the curse of dimensionality? Everybody's going to give you a slightly different answer. We don't really know what the curse of dimensionality is. And it turns out, we did a series of simulation studies where we chose, it just happens to be as many dimensions as I could have in my data, but we said, okay, well, let's say the true effect lies in some degree of dimensional space from 1-dimensional up to 59-dimensional, and then we analyze it either at a low dimensional or perhaps a too high dimensional. Well, what could go wrong? So basically, power tends to go up as long as the dimensionality of the trait is high. The more things that you actually analyze, the more power you have to find effects. And there is a disadvantage if you go beyond. So if you were to analyze a two-dimensional trait at 59 dimensions, you wouldn't do very well. Power would be very low. But these are fairly small effects. And then there's no problem with type 1 error or anything like that. So everything behaves well in that sense. So here's the actual analysis of the data. We're contrasting a 59-dimensional analysis with a 1-dimensional. We should take PC1, which is probably the easiest single dimension on which to find things. And this is just picking an arbitrary p-value here as our criterion of significance. And you can see there's a whole lot of stuff up here. Another feature of these data is there's a whole lot of regions of the genome that seem to be implicated. There aren't a few peaks that have a lot of signal at them. And there's far less if you do PC1. If you take a slightly more sophisticated approach to picking what's significant and what's not, so this is the Benjamini-Hochberg model, which models the distribution of p-values as a mixture of two populations. There's the uniform distribution of p-values where there really isn't an effect, and then a mixture, some distribution of p-values where there really is an effect, regardless of whether they cross. So that the estimate you get from that two mixture model is that there are almost 30% of the SNPs are inferred to have some effect for the MANOVA, the full dimensional analysis, and only about 5% are inferred to have an effect on PC1. If you then actually ask which ones are significant, we have almost 2,400 that are significant with the multidimensional analysis, and exactly 4 that are significant for PC1. We also have inversions in these data, which have been somewhat neglected in prior GWAS studies. 2 of them have very highly significant effects on wing shape. Now, there's a problem. Even though this is Drosophila, an outbred population of Drosophila, and there's less linkage disequilibrium than in many populations, there's still plenty of linkage disequilibrium. So what we did, we actually calculated the linkage disequilibrium between every pair of SNPs in the entire genome. This data is just for the ones that are significant by that multivariate one. And so the probability that a SNP is going to be correlated at r² 0.5, which we've done some simulations, that's where you begin to get confounding of the signal in this kind of design. More than 50% of the SNPs are highly correlated with something. And more disturbingly, for the rare ones, they're very likely to be correlated with something that is, well, see, this is a mistake. That should be 100 kilobases away. So there's a whole lot of stuff that's correlated with stuff on other chromosomes. And basically, we call this rarity disequilibrium. There are 2.5 million SNPs, and there's only so many ways you can take, say, 5 SNPs and put them in 184 lines, and with 2.5 million, they become correlated just by chance. So we had to try and take account of this, and what we did was we did a series of filterings to see which SNPs were relatively interpretable. Those that are not correlated with other SNPs, for example, they tend to be high minor allele frequency things. Those that, when you put them in a multivariate multiple regression, still retain their signal when you do this. So there's a series of these, and when we do this, we get down about 10% are sort of super interpretable. They're not in high LD with something that's so far away that it doesn't tell you something about the locus. Turns out that, like, the gene ontology analysis comes out essentially the same, regardless of whether you're using the full set or and we get anatomical development, morphogenesis, neurogenesis. There's the whole notch pathway, et cetera, is involved in forming the margin of the wing. So those are intimately related. These make good sense, as do these receptor activities, transcription factor binding, et cetera. So here's another advantage with multivariate inferences, is that when you do a validation, So a typical validation with a univariate is you would knock down the gene that you've implicated and you see, oh, does it or does it not have an effect on that trait? But in a high-dimensional space, you can not just ask, does it have an effect, but does it have an effect that points in the correct direction to either be against or in the same direction as the SNP. This R squared is a highly significant correlation here. This is a gene that formerly it's known to be involved in digestion, the development of the digestive system. It is expressed in the wing, and there's a cluster of about 6 SNPs that do that. And I'll point out that these visualizations, the movies, and these are what we call parrot plots. They're local expansions or contractions. Blue are contractions of that area of the wing inferred, and the reds are local expansions of that area of the wing. Now, we started to ask a question, and this I'm not really sure the answer. I'll show you some data here. So why is PC1 PC1? Why are some things more variable than others? And in principle, we split this into 2 possible explanations. One is if all SNPs have, say, the same effect size, regardless of the direction they point, PC1 could be PC1 because there's lots of effects that tend to point along PC1 as opposed to other directions. Or we could have direction be completely random, but the size of the effect is bigger if you point in PC1 than if you point in other directions. And so we first did this with a univariate analysis, and there's variation in how many hits we get. This is using a fixed p-value of 10 to the minus 6 for significance. There's a few less down here. This turns out to be an artifact that we've corrected with an inversion. But the effect size shows this very nice monotonic, nearly monotonic relationship where things that that are inferred for PC1 have bigger effects than those for other PCs. When we look at this in a multivariate way, there's a couple of ways to do this. This is a little bit of a hard slide to get your head around. So first of all, we took all the effect sizes of the significant SNPs. These were filtered for LD. And we split them into deciles. So smallest effects, biggest effects. And then here's the proportion that's explained, the cumulative proportion that's explained by PC1, 1 plus 2, 1, 2, 3, etc. And you can see that, at least in this 3-dimensional space especially, that the biggest effects tend to fall, be more on those high variance PCs. Here's another way to approach it, which is less clear. We basically took every effect that we estimated and we assigned it to one PC. Whichever the angle was the smallest, we say that's a PC1 vector if it's closest to PC1. The number is much higher in those first PCs. So there does seem to be a signal of the number of PCs that point in that direction. But you can see that the extremes here, at least, and to some extent the means are bigger if you point in that direction. But it's not nearly the clear result you got with the univariate analysis. So I'm open to suggestions on that. I don't really know what the truth of it is. But anyway, it's an interesting question to ask. So in conclusion, power is actually increased by a multivariate analysis. You're not going to lose much if you do that. Validation of effects is more informative. In other words, the direction of the vectors is something that's really interesting to look around. And then we do recapitulate the gene matrix pretty well with these SNPs, which of course kind of has to be true. But it seems to be both a signal of more effects on the variable dimensions and the bigger effects pointing along those dimensions. Thanks very much.